Now to the rescue of the Griffith experiment after 15 years of the Griffith experiment, okay, around in the year 1944. The Griffith experiment was done in 1928, okay, and the Avery Macleod and Mccarty experiment was done in 1944. Now they come in the rescue to the factor, okay, to investigate the factor, that what was the factor which transformed the cells from R to S, okay, from rough colony producing to smooth colony producing. What made the R cells produce capsule? This was very astonishing fact that the R colonies or the R cells were producing smooth or colonies containing capsules. It was done by Oswald Avery, Colin Macleod, okay, and Macleod Mccarty. What did they do? They subcultured the transformed cells again and again, okay. Now the R cells were transformed into S cells, but they had to investigate that whether the change was permanent or not, that whether in the next generation the S cell or the transformed S cells will again become R cells or not, okay. So to check that whether the effect or the Griffith effect was permanent or not, they subcultured the cells or the transformed cells again and again in nutrient agar. And what did they see? They see that the cells were producing smooth colonies in every generation, okay. In every generation they were producing smooth colonies. Now for the experiment part, what did they do was they took the S cells or the smooth colony producing cells in a medium, okay, then they centrifuged it and took out the pellet, okay, and they heat killed it. The heat killed the pellet, or the pellet was the cells, okay. We need to understand that the cells were in the pellet. After extraction of the polysaccharides and lipids, they were having the filtrate, okay. Now the filtrate was first treated with protease. The protease, what did the protease do? The protease degraded all the proteins, okay, and the filtrate was transferred to a flask containing of rough cells, okay, which were avirulent. But the filtrate, when it was transferred to the flask containing R cells, okay, transformation occurred, means the protein was not the genetic material, okay. It meant that the protein was not the genetic material, because though degraded it can transform, hence it was either between the RNA or the DNA, okay. Now in the next part, what they did, they took the remaining filtrate and treated it with ribonuclease. The ribonuclease degraded the RNA, okay. Now only DNA was present in here, okay, only DNA was present in the filtrate, and they took the filtrate and poured it in a flask containing R cells, okay, R2 cells, and hence we can see again that again transformation occurs. Then we can say that it must be DNA or other substance which may be present in along with DNA. These two may be the genetic material, okay. Now in the fourth installment, what did they do? They put deoxyribonuclease or DNase enzyme with the remaining portion of the filtrate, and as we can see, no transformation occurs. Hence we can conclude that only DNA is the genetic material which transforms the R cells to the S cells. We can easily conclude that, okay. Now this was it. This was the Griffith experiment and the Avery Macleod and Mccarty experiment explained. This is a very important topic for 5th sem in especially our BAMU, Baba Saheb Ambedkar Marathwada University, and all other universities. It comes in the 5th sem of genetics, okay, in BAMU.